So our next speaker is Senbai Kang, who's a PhD student at Ewa Szturek Lab at the University of Warsaw. He's going to tell us some interesting updates or extension on what David was talking about. His model SIEVE is doing joint inference of both phylogeny and SNVs from single-cell sequencing data.
Thank you, Magda. And also thank you, David. And built upon David's presentation, I'm going to show you our recently finished project, namely SIEVE, joint inference of single-nucleotide variants and cell phylogeny from single-cell DNA sequencing data. So this project is really a collaborative work among CONTRA. So from Nico Boxmüller, Monica, and David Posada, and Nico Birwinkel. Okay, I'm going to start. Our whole story starts with the well-known behavior, a phenomenon of intratumor heterogeneity, which is mainly driven by somatic mutations. As you can see in the picture, somatic mutations accumulate as time passing by, resulting in different tumor subclones possessing different genetic profiles. And this phenomenon poses significant challenges to diagnosis and clinical therapy of cancer. And the tool that people use to study it is phylogenetic inference, where we want to find the best phylogenetic tree that can explain data. In our project, the data we use is single-cell DNA sequencing data, which enables the reconstruction of cell phylogeny. And the specific data format that we use as input to our method is row read counts for all nucleotides. It helps reduce the uncertainties introduced by third-party random calling tools. However, the single-cell DNA sequencing data is highly error-prone. As you might know, it mainly because of the technical artifacts. For example, one of the alleles can be completely dropped out during amplification and never be sequenced at all, which is called egg dropout or ADO. And one of the alleles whose nucleotide can be amplified or sequenced into a completely different nucleotide. And also one big problem of the single-cell DNA sequencing data is abundant button missing data. So it is important that this kind of data requires a specific model to take as input row read counts and to properly account for technical errors. As David mentioned before, there are many methods or softwares for phylogenetic analysis. One of them is called BIS2. It has many advantages. For example, it is highly extensible. It already has many packages for the classical phylogenetic analysis, and it is also very user-friendly. But it also comes with many disadvantages. First of all, it always requires genomic sequences as input, and it is currently still incompatible with single-cell DNA sequencing data. So it is necessary to have a new package of BIS2 to bring to the platform tumor phylogenetic inference using single-cell DNA sequencing data. Now I'm presenting our model SIEVE, which is shorthand of Single-Cell Evolution Explorer. In a nutshell, it is an extension to statistical phylogenetic models. So our model first, it contains a statistical phylogenetic model, which models the evolution of single cells. If you look at this picture, this is a probability graphical model that we developed. So the statistical photogenetic model is to the left of the panel. Inside this orange dotted frame, it contains a tree topology, a branch length, as well as a instantaneous rate matrix, which is defined by us. It contains 4 possible genotypes, 0, 0, 0, 1, 1, 1, and 1, 1 prime. So inside of those genotypes, 0 represents reference nucleotide, 1 is the alternative nucleotide, and 1 prime is another alternative nucleotide which is different from 1. Our model also contains another submodel, let's say, which is a model of row rate counts. And this is also the reason why we call it extension to statistical phylogenetic models. In this model of row rate counts, it accounts for the technical errors, and it is represented in the picture to the right side of the graphical model, inside the blue dashed frame. So basically, we represent our input data, row rate counts for all nucleotides, as Dij, which is the combination of Mij and Cij. Mij here is a vector of read counts for 3 alternative nucleotides in descending order, and Cij is sequencing coverage. Altogether, the probability of observing Dij can be decomposed into 2 different distributions. One of them is Dirichlet multinomial distribution, another one follows a negative binomial distribution. We choose these 2 distributions because they can very well capture the over-dispersion inside the single-cell DNA sequencing data, which is highly noisy. Okay, there are more features about our model. For example, we perform acquisition bias correction for accurate branch lengths during the inference, and our model is implemented as a package of BIS2. So now there are some more results from the simulation study. We first compare the tree branch lengths using the criterion of rooted branch score distance from the true tree. So first of all, in our simulation, we have 3 main variables. First one is the mutation rate. We have 3 categories. And second one is number of cells. We have 40 cells or 100 cells. And the third one is the coverage qualities, where we vary the mean and variance of the sequencing coverage per allele. And altogether, it gave us 18 possible combinations altogether. And in terms of the tree branch length estimate comparison, we compare with SIEVE as well as Senbai, and it's quite obvious that we achieve the lowest value here. By the way, the gray dashed line here is the optimal value of this metric. This is because we are the only method among those 3 methods that perform acquisition bias correction, and the resulting branch length is measured in different units. For example, for SIEVE, it is measured in the number of somatic mutations per genomic site, but for Senbai and SIEVE, they are measured by the number of somatic mutations per SNV site. There are also some parameters that we are interested in. For example, the effective sequencing error rate, which accounts for both amplification error and sequencing error. So in this figure, it is also quite obvious that we are the most robust and most accurate model of all the similarity scenarios compared to others. Another parameter which is important is error rate, and the conclusion from the previous slide still holds. Okay, now some more results from the variant calling section. So we first compare the single mutant genotype calling, which is mainly the performance in calling genotype 01, and we compare it in two different metrics, so F1 score and false positive rate. So in the F1 score, it is also quite obvious that we can achieve the highest F1 score among all the methods and the simulation scenarios, and it is almost very close to 1. And for the false positive rates, our methods as well as the monovar can almost achieve 0 false positive rates, which means it is highly accurate to call genotype 01. Okay, next we focus on the double mutant genotype calling, where we call genotype 11 and 11 prime. In terms of the F1 score, we will compare with monovar here, and it is, we perform much better than monovar, and this is mainly comes from the performance in the precision part. Because as you know, the F1 score is mainly consisted of recall and precision, and in terms of recall, we are slightly higher than monovar, but in terms of precision, we are much higher than monovar, and resulting in a much higher F1 score. And in terms of false positive rates, you can see that SIEVE can always achieve zero false positive rates in terms of these two genotypes, and for some reason monovar has higher false positive rates. It means that in the variant calling results of monovar, if you see, there are some false positives of the genotype 11, which is truly not, not this genotype. Okay, there's more results from the real data. First of all, it's the colorectal cancer. We nicely acquired from David's lab. Here in panel A, this is the heat map of the SIEVE variant calling results, and here is monovar variant calling results. And it's clear that we don't have any missing entries because using phylogenetic tree we show here, we can fill up those empty places, and we do call very few of those two double mutant genotypes, while for monovar it calls a lot, actually genotype 11. From the previous slides, it might mean that some of those 11 genotype called by monovar can be false positives. In terms of the tree, first of all, we group those cells from different anatomical sites together, which is very nice. And we also mapped those non-anonymous genes to the branches. And for this data set, we only observe one type of mutation event, which is single mutation. But for the next one, it will be quite different. For this data set, we observe a lot of other types of mutation events, which is not single mutation. For example, here we have double mutation, and here we have some single back mutation, and we also observe some parallel evolution. And this is actually one more feature of our method, that we can categorize different mutation events into different types. And also for this data set, we observe a lot of double mutant genotypes compared to the previous one. Okay, to summarize, SIEVE takes as input row read counts for all nucleotides. It includes a novel statistical phylogenetic model and a model of row read counts. It can reliably construct cell phylogenies, estimate parameters, and covariance, especially double mutant genotypes using MSMC. It also can differentiate different mutation events, and it is implemented as a package of BIS2, which is compatible with other features and existing packages of the framework. And finally, I didn't cover every detail of our method, so if you're interested, please read this paper. It's on bioRxiv now. Finally, I'd like to acknowledge my supervisor, Eva, which is super supportive, and our fantastic collaborators from Professor Nico Bierwinkel's lab and Professor David Pozzola's lab. And also, Eva asked me that to mention that we have currently some postdoc positions in her lab, and if you're interested, please check his link. I personally very recommend our lab. Okay, with that, I'd like to thank you for your attention. I'm happy to take questions. Yes, please.
Can you describe a bit more in detail how this part of the model where you have a negative binomial and then conditioned by the value, what you get from that, you have a de-risk here?
Mm-hmm. So the data itself, it contains two parts, actually, MIJ and CIJ. And I mentioned MIJ is read counts of 3 alternative nucleotides. CIJ is sequencing coverage. So the probability of observing DIJ can be decomposed further into 2 components. So first is the probability of MIJ given CIJ. Next is the probability of CIJ itself.
Right.
Yeah.
You have a negative binomial. There's some sort of, if you only observe the count, right?
Yeah. Actually,
unless you infer some parameter for that negative binomial, or it depends on a copy number or something.
Yes, that's right. Yeah, it's actually inside. It's the number of sequenced alleles, which plays part inside. So we actually have some assumptions about the mean and variance of negative binomial distribution. We use a nice model to link them together, and we have 2 free parameters, which is the mean and variance of sequencing coverage per allele, which are NERD inside of the MSMC.
Okay. So then partly, is the copy number evolving over the tree, or is it just there?
It's not copy number.
It's not copy number, it's the number of alleles amplified in whole genome amplification.
Yeah.
The number of templates.
We don't model here
The evolution.
the copy number evolution, and we assume you have a deployed genome,
Deployed, yeah.
but you can have the other.
So there is just some number of alleles, which of
Yes.
course is like a copy number.
Yes, exactly.
But then how do you get the Dirichlet from conditioned by that?
Yes, I mean, given the conditional on the sequencing coverage, we have 3 values actually here. The read counts of, I mean, for 3 alternative nucleotides, and we have the sequencing error, I mean, effective sequence error rates inside. So each nucleotide will have a expected rate.
Okay, so it's a bit of an extension of the earlier model that Nico.
Yes, that's right. Mm-hmm. Sir?
Can you explain a bit more how, and also first why you do detect the double mutant genotypes, and why monovar doesn't exactly?
Mono, okay.
Or even how do you try to detect double mutants using monovar?
Okay, so I think
No, you're at the right slide.
maybe you can use these slides to explain to you better.
Data? From the synthetic data?
So for my question,
Yeah.
how do you like,
So I think we can go back to this model, right? So GIG here, this is a bridge between the statistical phylogenic model and the model of row read counts. And it actually represents the genotype of the cell J at site I. So genotype itself can be one of the 4 genotypes. So during the inference process, we actually try to infer the maximum likelihood genotypes of that specific cell at that specific site. So that's the reason why we can differentiate those 2 different genotypes.
and that's why, or monovar, it's the monovar.
Okay.
It's because of the allele
Why
dropout.
the monomer doesn't see
Yeah,
it?
it's because of the allele dropout.
Mm-hmm. Oops. Okay.
It's because of the model of
Oh.
Of SIEVE. No?
Yes, I think it's primarily because we use the tree, use the phylogenetic relations. Yes.
But then, how does it even see it? Like, I would have thought that you cannot see any of those, like any of the double mutant genotypes using monovar.
It's actually one of our reports a lot of double mutations.
Okay.
Although I think most of them could be false positives because based on these plots, right, it has some non-zero, like 2% false positive rates. Well, for us, it's like always 0. So
monovar doesn't use a phylogeny.
yeah.
So the phylogeny helps you identify something that might be a double mutant. You say, no, it has to be a allele dropout because it's next to, it's related to other genotypes. And also the coverage model is different. This is much more sophisticated.
Yeah.
Okay. Thank you.
Thank you.